I'm Mike Clark from the University of Melbourne, and I'm going to talk a bit about some of the software tools that we have created to allow us to analyze RNA isoform expression using nanopore sequencing, and hope that perhaps some of these tools from the IsoMix toolkit might be useful for your lab as well. So something that motivates a lot of our work is the fact that almost all human genes express multiple RNA isoforms. I show an example of this here in the cartoon form, where we have a gene with 5 different exons that gets transcribed into a pre-mRNA, and then a process called splicing occurs where the introns are removed and the exons are kept and spliced together to create a mature mRNA. Now, there is also a process called alternative splicing, which modifies which exons are included in the mRNA. An example of this is shown here, where exon 3 has been skipped and removed. Why this can be important is that alternative isoforms can create RNAs and also proteins with varying sequence and therefore varying function. There are 2 examples shown here, and we are very interested in trying to understand the identity, expression and function of different RNA isoforms in the brain, in risk genes for neuropsychiatric disorders, and also in how they can cause rare disease. We've been using long-read nanopore sequencing for this because long reads are really useful for identifying expressed isoforms. There's an example here. We're using a cartoon again of a gene with 5 exons. When we do our long-read sequencing, we are often able to sequence the entire mRNA or cDNA in one go, and therefore we can see all of the exons that are present in each RNA and therefore understand what isoform is present. We can also quantify the isoforms better this way as well. And we've been using nanopore sequencing for several years now, and what often we've found that to answer the questions we want to answer, there aren't always existing tools available. So we've ended up creating quite a few ourselves, and we've created what we call the IsoMix toolkit, which is present at isomix.org. These tools include NanoCount, which is for the quantification of RNA isoforms from direct RNA sequencing, Blaze, which is a new tool to allow you to do long-read only single-cell sequencing, which I'll be talking about in a couple of minutes, NanoSplicer, which uses RNA squiggles or raw sequence from the nanopore output to correct and identify splice junctions, Discoland, which is a new tool for the discovery and annotation of transcript isoforms from amplicon sequencing, and also the isoform visualizer or isoviz, which is another new tool for RNA isoform visualization. So let's talk first about Blaze. This work was spearheaded by Sefi Prauer and Upei Yu, 2 PhD students in the lab. Sefi has a poster on Blaze here at the conference if you're interested in more information. So for the last 10 or so years, there's been a lot of activity in doing single-cell RNA sequencing because it allows you to understand cell-specific expression patterns. However, most single-cell RNA sequencing only sequences the very 3' ends of RNAs. On the other hand, long-read single-cell RNA sequencing can allow you to understand cell-specific isoform profiles. The key step in RNA sequencing is identifying what cell each of your sequencing reads came from, and this is achieved through something called a cell barcode, which is present in each read. Now, previously, most people who want to do long-read single-cell sequencing have used matched short-read sequencing to identify the barcodes. Basically, it worked like this. You had your barcoded reads, and you performed short-read sequencing to create a list of barcodes for each cell. You then did long-read sequencing on the same sample, extracted the putative barcodes that would often have sequencing errors in them, which meant it was difficult to understand exactly what barcodes or cell barcodes they were, and then you do a matching process to assign each of the cell barcodes to your putative barcodes, and in that way you could work out which cell each of your long reads came from. With our new Blaze tool, we've eliminated the need to do any short read sequencing at all. We can now accurately detect the cell barcode list directly from the long-read sequencing. This saves both time and money, and as we actually also found, gives you more accurate results. And I'll give an outline of why that is here. So here's some of the sequencing we did. It was on 5 cancer cell lines. We knew what cell lines were present, so we know that when we sequence them, we should get only those 5 cell lines, and when we cluster them, they should form 5 different clusters, one for each cell line. This is what we see with Blaze. When we did short read sequencing and then used the tool Cell Ranger to identify the barcodes and hence the cells, we found an extra cluster of cells that shouldn't be there. And when we used the previous version of ONT's sockeye tool, we found a large number of additional clusters that shouldn't be there, suggesting that we have found barcodes, or these tools have found barcodes, and therefore cells that aren't actually present in the sample. Why did this happen? Well, one of the reasons is that in the case of Cell Ranger, from the short reads, some of what were real barcodes and therefore cells in the short read dataset didn't actually exist in the long read dataset. So therefore, some of these extra barcodes don't represent real cells, they represent empty droplets. You'll notice we did not find any of these with Blaze, but we did find quite a few of them with Sockeye as well. Sockeye also appeared to have a separate problem where it would identify barcodes with sequencing errors as true unique barcodes. However, these are actually false positives, and we're showing all of these false positives here. These can be a problem for several reasons, and one of them is that because the barcodes are very similar to real barcodes, these false positive cells can be assigned reads and sometimes actually look like real cells, although they are not. With the accurate detection of the cell barcodes from Blaze, we were able to get good biological signal from our long-read single cell data. So here I'm showing some barcodes and therefore cells that have been clustered. They come from stem cells that were differentiating into cortical neurons. What we could see was that one of the clusters was expressing neuronal transcription factors and various markers that identify developing neurons. So this gives us good confidence that with our Blaze technique, we're able to identify the relevant barcodes and cells and the biological signal in the sample. Alright, I'm going to now move on to Discoland for a couple of minutes. This is for detecting isoforms from amplicon sequencing. This work has been led by Ricardo and Schwaiter, and Rick has a talk about this project in the breakout session on human transcriptomics. So one thing our lab is really interested in is risk genes for neuropsychiatric disorders. We know that most neuropsychiatric disorders have a strong genetic component, and hundreds of risk genes have been identified. At a global level, we know that these genes tend to mediate risk through changes in their expression and splicing patterns. But at the individual gene level, we generally don't know how a specific gene is contributing to disease. Therefore, my lab, we were really interested in trying to better understand the splicing patterns of risk genes in brain, to then, as a first step for understanding how they're causing disease. We looked at 7 regions of post-mortem human brain from 4 individuals, and we did a long-read PCR so that we could amplify out pretty much the entire mRNA of each gene. We then did the long-read sequencing and then isoform detection and quantification with our tool Blaze to identify both known and novel isoforms. We looked at the isoform profiles for 31 neuropsychiatric risk genes. These genes were risk for schizophrenia, depression, bipolar, and autism. In total, we found over 900 different isoforms from these 31 genes. The vast majority of them were novel. When we have a look at each gene and ask how much of its expression was novel isoforms versus known, we can see that the results are very varied. However, we can also see that despite 2 or so decades worth of annotation of human genes and transcriptomes, there were quite a few genes where a lot, if not the majority, of their expression actually came from novel isoforms. An example of this is the schizophrenia risk gene, ATG13. Now I'm showing here actually an IsoVis plot to show the isoforms and their expression. So up here we have the canonical transcript, and then we have in this isoform stack all of the transcripts that were found in our sequencing, and over here we have a heat map of transcript expression or isoform expression in each sample. We show exons as boxes and coding sequence are the thicker boxes. What you can perhaps immediately see is that by far the most highly expressed RNA isoform in brain for ATG13 was in fact novel. We can also see that there are alternative splicing hotspots in both the 5' UTR region and also within the coding sequence. So this combination of Discoland and also IsoVis allows us to really thoroughly understand the splicing profiles of our genes of interest, and of course could be applied to any particular genes of interest. So in summary, with the IsoMix toolkit, we've created a series of easy-to-use tools to identify, quantify, and visualize isoform expression in bulk and in single-cell samples. We're using them for a large range of areas that are of interest to our lab, but they're certainly widely applicable to questions that almost any lab has. These tools can be found at isomix.org, and if you're interested in these areas of research, I really hope you'll give them a go, and I hope that they help your work. I'd just like to acknowledge all the members of my lab who have contributed to these various projects, as well as our collaborators and our funding agencies, and you for listening. Thank you.